Hello, everyone. My name is Ning Zhang. I'm a postdoc at Paczewski Lab in the Department of Pharmacology. My talk today is knocking E150K opsin mutant mouse model for autosomal recessive retinitis pigmentosa, features structural disorganization of photoreceptor internal membranes. I'd like to mention that the background image has recently won the Art of Science in the School of Medicine and showing the activated Mueller cells in the E150K opsin mutant mouse. And retinitis pigmentosa is a group of retinal degenerative diseases that are characterized primarily by the loss of rod photoreceptor cells, and it affects over 1 million individuals in the whole world and estimated 100,000 people in the United States. Based on the pattern of inheritance, they are autosomal dominant, autosomal recessive, and X-linked retinitis pigmentosa. The symptoms include night blindness and loss of peripheral vision, causing tunnel vision as seen in the right picture here. Unfortunately, by far, there is no treatment for this blinding disease. Retinitis pigmentosa is a heterogeneous group of hereditary retinal disorders caused by over 50 different genetic mutations of the photoreceptor proteins. And among them, more than 100 identified mutations in rod opsin genes have been reported to associate with retinitis pigmentosa, and most of them cause autosomal dominant form of this disease. Only a few of them cause autosomal recessive form of the disease. So E150K is the first reported missense mutation in rod opsin gene associated with autosomal recessive form of this disease. As shown in 3D and 2D rhodopsin structure, glutamic acid 150 is located in the C-terminal region of the second cytoplasmic loop, embedded at the edge of the phospholipid bilayer. The E150K mutation redistributes the charge of rhodopsin and causing the positive charge accumulation at the membrane surface of cytoplasmic side. To better understand the pathogenesis and design the possible treatment for retinitis pigmentosa, we generated the E150K knock-in mouse. In this mouse, the wild-type rhodopsin allele was replaced by E150K mutation rhodopsin, and with the FLoxP neomycin box inserted in front of the axon 2. And later, the nail box was removed by Cre-expressing vector, and then the final knock-in allele contains E150K mutation and 44BP FLoxP residue in front of the axon 2. The mutation was confirmed by genotyping. The KK here denotes homozygous, and the EK here denotes heterozygous E150K mouse. We can see in wild-type mouse, we have 262 BP PCR products, and in homozygous mouse, we have 306 BP PCR products. And we also confirmed with the direct sequencing of the cDNA. So G2A alteration shown here is observed in the E150K mutant mice. And we did a morphological study of the E150K eyes. At 1 month of age, we can see a shortened outer segment and outer nuclear layer of homozygous mouse. And by the age of 4 months, almost all the photoreceptor degenerated in homozygous mouse. And we plotted the thickness of outer nuclear layer during aging. And we can see there is a quick decrease in outer nuclear layer thickness in homozygous mouse, indicating the rapid degeneration in homozygous mouse. And we, surprisingly, we also see the decrease in outer nuclear thickness in heterozygous mouse, and indicating a slower retinal degeneration in heterozygous mouse. So if this is also the case in the human, we need to re-evaluate this disease and examine the heterozygote individuals periodically. And we did a transmission electron microscopy for the E150K mouse, and we can see here in the homozygous mouse, there are abnormal phagosomes inside the RPE cells, indicating the improper disc shedding in the E150K mouse. Also, we can see disorganization of the outer segment in the homozygous E150K mouse. And we can see this inclusion body with a thick coating outside. For older mouse, we can see also the abnormality in the heterozygous mouse. There are deposit in the subretinal space, and for homozygous mouse, the outer segment is the outer segment structure is gone, and sometimes we can see the pigmented deposit at the homozygous E150K mouse. And we measured the function of E150K rods, and this is a single-cell suction recording. The mutant rods can sense light, but with the lower magnification. It's mainly due to the shortening of the outer segment. And we also looked at the kinetics of the photoresponse, and we can see the broaden of the peak and the quicker recovery of the photoresponse. And the recovery time from saturating flashes also accelerated in the homozygous mutant mouse, indicating the accelerated transducing PDE inactivation in the mutant mouse. And we can also see the dark noise increased in E150K homozygous mouse. Here, the low frequency noise is increased. And the thermal stability at a 60 degree of rhodopsin is also accelerated. It's less stable at the first 45 minutes. And we bred E150K knockout mouse with L-ret knockout mouse to eliminate the 11-cis retinal. As we can see here, the genetic depletion of 11-cis retinal somehow partially rescued the photodegeneration in the E150K mouse. So in summary, we generated a knocking E150K opsin mouse model which features progressive photoreceptor degeneration mimicking retinitis pigmentosa in human. And we saw disorganization of rod outer segment and aberrant phagocytosis of disc membrane in RPE cells in E150K mice. And the E150K mutant rhodopsin is capable of phototransduction, but due to disorganization, out segment and photoreceptor degeneration, overall vision function is impaired. And the E150K mutation in rhodopsin altered the binding with 11-cis retinal, causing less thermal stability, increased the spontaneous activity of rhodopsin. This may contribute to the increased dark noise in E150K rods. And the E150K opsin appears more stable than its counterpart rhodopsin, inducing a milder retinal disease phenotype. So all this indicate a novel mechanism of retinal degeneration in rhodopsin mutant mouse. So I'd like to thank my mentor, Dr. Chris Paczewski, for his continuous support of the project, and thank Beata for the biochemistry experiment, and Debashi for the EM study, and Satsumi for technical support, and Tivada for the 3D rhodopsin structure. And this work is collaborated with Sasha in Dr. Kafalov's lab, and I'd like also thank Maida Lab for the help of in vivo imaging, and I also want to thank the NIH grants to support this research. Thank you for your attention.